The Blood That Never Died, Neanderthals and the DNA Truth. Geneticists found, when they sequenced the first complete Neanderthal genome in 2010, that the creature the 19th century had dismissed as a brutish evolutionary dead end was something far more complicated. A close relative whose DNA still circulates in the bodies of most people alive today, and whose story, told through ancient bone and modern sequencing technology, dismantles one of the most persistent myths in the history of science. Neanderthals evolved in Europe and Western Asia approximately 400,000 years ago and survived until roughly 40,000 years ago, a span of time nearly 40 times longer than all of recorded human civilization. During that period, they adapted to some of the harshest conditions any hominin has ever encountered. Their bodies reflect those conditions directly, broader and more muscular than modern humans, with barrel-shaped chests that maximized lung capacity, large nasal passages that warmed and humidified freezing air before it reached the lungs, and a skeletal robustness that speaks to lifetimes of intense physical demand. Their brains were, on average, slightly larger than those of modern Homo sapiens, a fact that was known from the fossil record long before anyone could sequence their DNA, and that made it difficult to maintain the traditional picture of Neanderthals as cognitively inferior. Brain size alone does not determine intelligence, and the internal organization of the Neanderthal brain differed from ours in ways that are still being studied. But the sheer volume was there, and it demanded explanation. The tools they made, the Mousterian stone technology documented at hundreds of sites across Europe and Western Asia, show a consistent, sophisticated approach to working stone that required planning, skill transmission across generations, and the kind of standardized technique that implies teaching rather than individual improvisation. Ancient DNA researchers found, when Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology published the first draft Neanderthal genome in the journal Science in May 2010, that modern humans outside of Africa carry between 1% and 4% Neanderthal DNA, a finding that ended a decades-long debate about whether Neanderthals and modern humans had interbred, and that earned Pääbo the Nobel Prize in Physiology or Medicine in 2022. The challenge of extracting usable DNA from bones 40,000 years old was immense. DNA degrades over time, broken into shorter and shorter fragments by heat, moisture, ultraviolet radiation, and microbial activity. Ancient samples are typically contaminated with bacterial DNA that vastly outnumbers the human genetic material of interest. Pääbo's team developed laboratory protocols for working in ultra-clean conditions and computational methods for distinguishing genuine ancient human DNA from contamination, drawing on the characteristic chemical modifications that accumulate in DNA as it ages. The source material for the initial genome was bone powder from 3 Neanderthal specimens from Vindija Cave in Croatia, dating to approximately 40,000 years ago. The genetic data showed conclusively that the Vindija Neanderthals shared more DNA with non-African modern humans, Europeans, Asians, and people from the Americas and Oceania than with sub-Saharan Africans. The only explanation consistent with this pattern was interbreeding. At some point after modern humans left Africa, but before they spread across the rest of the world, they encountered and produced offspring with Neanderthals, and those offspring contributed to the lineage of everyone alive today outside Africa. Archaeological evidence found at Shanidar Cave in the Zagros Mountains of northern Iraq, excavated by American archaeologist Ralph Selecki beginning in 1957, produced skeletal remains of at least 10 Neanderthal individuals, spanning a period from roughly 70,000 to 45,000 years ago. And one of those burials generated a finding that changed the way scientists understood Neanderthal emotional and social life. Shanidar 4, as the burial is catalogued, was an adult male whose remains were associated, in the original excavation report, with large quantities of flower pollen concentrated around the body. Selecki interpreted this as evidence of deliberate flower burial, that the Neanderthals who interred this individual had placed flowers as an offering or expression of grief, implying emotional complexity and possibly spiritual belief. The finding was widely cited for decades as the most direct evidence of Neanderthal symbolic behavior. The flower burial interpretation has since been contested. A subsequent study suggested that the pollen concentration might have resulted from burrowing rodents carrying flowering plants into the cave, rather than from deliberate placement by Neanderthals. The debate is not fully resolved, and the original interpretation has not been definitively disproven. It remains a live scientific question. What is not contested is a separate finding from Shanidar. Shanidar 1, an older male, had survived to an advanced age despite a severely withered right arm, significant hearing loss in at least one ear, and multiple healed injuries, suggesting a life of accumulated physical trauma. His survival required assistance. Someone had to have provided him with food and care. This is unambiguous evidence that Neanderthals looked after their injured and elderly, a behavioral pattern that implies social bonds of considerable depth. Dating evidence found, when researchers applied uranium-thorium dating to mineral crusts overlying cave paintings at 3 sites in Spain, La Pasiega, Ardales, and Maltravieso, established that at least some of the paintings were created more than 65,000 years ago, placing their creation at a time when modern humans had not yet arrived in Europe. The study, published in Science in 2018 by a team led by Dirk Hoffmann of the Max Planck Institute, reported minimum ages for painted and engraved features, including red and black geometric shapes, hand stencils, and animal figures. The oldest dated feature, a red ladder-shaped symbol in La Pasiega cave, had a minimum age of 65,000 years, predating the earliest confirmed modern human presence in Europe by approximately 20,000 years. If the dates are correct, the artists were Neanderthals. The findings generated significant scientific discussion. Some researchers questioned whether the uranium-thorium method, which dates the calcite crust over the paintings rather than the pigment itself, reliably reflects the age of the art. The debate continues, but multiple independent analyses have supported ages in the range that would require Neanderthal authorship for at least some of the Spanish cave art, and the broader evidence for Neanderthal symbolic behavior has strengthened substantially in the past 2 decades. Eagle talons perforated and strung as jewellery, recovered from Krapina cave in Croatia and dated to approximately 130,000 years ago, predate modern human arrival in Europe by tens of thousands of years. Pigment use, red ochre and black manganese dioxide, has been documented at Neanderthal sites across Europe. The Divje Babe object, a cave bear femur with aligned holes found in Slovenia and dated to approximately 60,000 years ago, has been proposed as a flute, though whether the holes are man-made or the result of carnivore gnawing remains disputed. Genetic analysis found, when researchers sequenced the FOXP2 gene from Neanderthal DNA, recovered from specimens at El Cidrón cave in Spain, that Neanderthals carried the same 2 mutations in this gene that distinguish modern humans from chimpanzees. Mutations associated with the fine motor control of speech and with the neural circuits involved in language processing. FOX2 is sometimes described, imprecisely, as a language gene. It does not encode language itself, but is essential for the precise muscular coordination that spoken language requires, and for aspects of procedural learning that underlie both language acquisition and complex motor skill development. Individuals with mutations that impair FOX2 function have severe difficulties with speech and language. The fact that Neanderthals shared the human form of this gene suggests they had at least the neurological substrate for language. The hyoid bone, the small horseshoe-shaped bone in the throat that supports the tongue and is essential to the production of a wide range of speech sounds, has been recovered from Neanderthal remains and found to be morphologically indistinguishable from the modern human hyoid. Computational modeling of the Neanderthal vocal tract, based on skeletal evidence, suggests they were capable of producing a full range of vowel sounds, though their voices may have been higher pitched than modern human voices due to differences in the size and shape of the resonating spaces. Reconstructing whether and how Neanderthals used language is ultimately beyond what the available evidence can definitively establish. But the genetic and anatomical evidence is consistent with a species capable of complex vocal communication, and inconsistent with the traditional portrait of a creature limited to grunts and gestures. Dental calculus analysis found, when researchers examined mineralized plaque deposits on Neanderthal teeth from El Cidrón Cave in Spain, evidence of a diet and a relationship with medicinal plants that was significantly more sophisticated than the meat-centered picture of Neanderthal subsistence that had dominated the field for most of the 20th century. The 2017 study, published in Nature by a team led by Laura Weyrich at the University of Adelaide, extracted ancient DNA and chemical compounds from the dental calculus of 5 Neanderthal individuals from El Cidrón. The results showed that the El Cidrón Neanderthals consumed a diet that included cooked plant material, starchy vegetables and wild grasses, alongside meat. One individual showed no evidence of meat consumption at all, with a diet consisting entirely of plant foods, including mushrooms, moss, and tree bark. More striking was the finding related to one individual who showed skeletal evidence of a dental abscess and intestinal parasite infection. The calculus from this individual contained traces of poplar bark, a natural source of salicylic acid, the active compound in aspirin, and of penicillium mold, a natural antibiotic. The researchers proposed that this individual may have been self-medicating with plant materials whose therapeutic properties were known within the group. The El Cidrón findings also recovered Neanderthal nuclear and mitochondrial DNA from the specimens, allowing the researchers to determine that all 5 individuals were closely related, consistent with a small family group. The mitochondrial sequences showed that 3 of the individuals were maternally related, a result that illuminates the social structure of a Neanderthal community at a level of biological specificity that no behavioral analysis alone could achieve. Population genetic modeling found, when applied to the ancient DNA record of late Neanderthal populations across Europe, evidence of a gradual and accelerating decline in genetic diversity, a biological signature of a population in trouble long before modern humans arrived in sufficient numbers to pose any competitive threat. Neanderthals were never a numerous species. Estimates of their total population at any given time range from around 10,000 to perhaps 70,000 individuals, spread across a vast geographic range from Western Europe to Siberia. At those population sizes, random genetic drift, the accidental loss of genetic variants in small groups, has a powerful effect, and the ancient DNA record shows the effects of that drift in the reduced genetic diversity of Neanderthal specimens from the last phase of their existence. The OASA-1 individual from Romania, analyzed by Chalmei Fu and colleagues in 2015, carried between 6 and 9% Neanderthal DNA, substantially more than any modern human population. and the analysis suggested that this individual had a Neanderthal ancestor within 4 to 6 generations. The finding documents that interbreeding was still occurring in Europe as late as 40,000 years ago, in the period when Neanderthal populations were in their final decline. Gibraltar's Gorham's Cave complex preserves evidence of Neanderthal occupation as recently as 28,000 years ago, making it the most recently dated Neanderthal site yet confirmed. By that point, Neanderthals had disappeared from mainland Europe and most of their former range. Whether the last populations died out from competition, disease, absorption into the modern human gene pool, or some combination of all three, remains an open question. What is clear is that they did not disappear without a trace. They dissolved biologically into us. Genomic research published across multiple journals since 2010 has identified specific Neanderthal-derived genetic variants in modern human populations and traced their effects on health, physiology, and disease susceptibility, establishing that the Neanderthal genome is not merely a historical curiosity, but an active biological presence in billions of living people. The immune system carries some of the clearest Neanderthal genetic signatures in modern humans. Several variants of HLA genes, the major histocompatibility complex genes that govern immune response, are of Neanderthal origin and are present at significant frequencies in non-African populations. These variants may have helped modern humans adapt to the Eurasian pathogen environment that Neanderthals have been navigating for hundreds of thousands of years, providing immunity to diseases that modern humans had not previously encountered. A study published in Nature in 2020 by Hugo Zerberg and Svante Parbo identified a segment of chromosome 3, inherited from Neanderthals, as the strongest genetic risk factor for severe COVID-19 outcomes identified in the genome. The same Neanderthal-derived segment that provides elevated risk for severe respiratory illness from SARS-CoV-2 may have conferred advantages against other respiratory pathogens in the prehistoric environment. Natural selection operates on the average fitness across all conditions encountered. A gene that was beneficial across hundreds of thousands of years of Neanderthal and early modern human life can become a liability under novel disease conditions. Other documented effects of Neanderthal DNA in modern populations include contributions to skin pigmentation, hair texture, sleep chronotype, the tendency toward being a morning or evening person, and elevated risk for certain allergic and autoimmune conditions. Archaeological and genetic evidence found at multiple Neanderthal sites across Europe documents a range of behaviors that, taken together, present a picture of Neanderthal social and ritual life that is more complex and more varied than any single site or line of evidence alone could support. At Krapina Cave in Croatia, the site that also yielded the eagle talon jewelry, researchers found hundreds of fragmented Neanderthal bones bearing cut marks in patterns consistent with defleshing. The original interpretation, proposed in early analyses, was that the Krapina Neanderthals practiced cannibalism. More recent analysis has proposed an alternative, that the cut marks reflect a mortuary practice of removing flesh from bones before secondary burial or disposal, a ritual treatment of the dead rather than consumption of the living. Both interpretations are consistent with the physical evidence, and neither can be definitively established from the marks alone. The most genetically significant Neanderthal individual discovered in recent years was found not at a Neanderthal site at all, but at Denisova Cave in Siberia. A bone fragment, identified as belonging to a female individual, designated Denny in published accounts, was found on genetic analysis to be the direct offspring of a Neanderthal mother and a Denisovan father, the first confirmed first-generation hybrid between two archaic human species. The finding, published in Nature in 2018, confirmed that the groups we identify as distinct species were not reproductively isolated from each other, and that the boundaries between them were biological gradients rather than hard barriers. The old man of La Chapelle-aux-Saints, a Neanderthal individual recovered from a French cave site in 1908, was elderly, had lost most of his teeth, and showed severe arthritis throughout his skeleton. His survival to old age in that condition required sustained care from his community. Scientific synthesis, found across the past 15 years of Neanderthal research, presents a portrait of our closest evolutionary relatives that is more human, more complex, and more relevant to understanding ourselves than anything the scientific community expected when Svante Pääbo began his work on ancient DNA in the 1980s. The revision in the Neanderthal narrative is one of the most complete in the history of paleoanthropology. In the span of roughly 50 years, the scientific consensus moved from a creature viewed as a separate, inferior species that modern humans replaced, possibly through direct competition or conflict, to a population that interbred with modern humans, contributed meaningfully to the modern human genome, created art, used medicinal plants, cared for their injured and elderly, and may have developed rudimentary language. Almost none of this was anticipated before the ancient DNA evidence began accumulating. Research published in 2021 by Alison Muotri's lab at the University of California, San Diego, introduced Neanderthal-specific variants of genes involved in brain development into human stem cells, and grew miniature brain organoids, tiny three-dimensional cell clusters that develop some features of early brain tissue. The Neanderthal variant organoids developed differently from control organoids in measurable ways, providing a window into potential differences in neural development between the two species that no amount of fossil analysis could have opened. The Neanderthal genome has given us something the fossil record alone could never provide: a direct biological connection to a species that lived and died before any written record existed. Their DNA is in us, not as contamination or accident, but as inheritance, carried forward because it was useful, because the people who carried it survived and had children. Neanderthals did not disappear. They became part of what we are.